The procedure performs dietary restriction on the standard agar-based plate in C. elegans to study its effect on longevity, reproduction, and metabolism. This is accomplished by first accurately measuring the concentration of the bacteria that will be used as a food source. The second step of the procedure is to properly dilute the bacteria culture into suitable concentrations. Then prepare agar-based SDR plates with killed bacteria. Finally, set up SDR experiments with an age-synchronized population of worms. Results of progeny production and lifespan measurements can show delayed and reduced progeny production as well as increased mean lifespan.
The main advantage of this technique over existing methods, like dietitian in arsenic medium, is that all the experiment will be done on a standard solid agar-based plate that is used by most worm laboratories.
For dietary restriction experiments in C. elegans, establish separate calibration curves for each bacteria strain used as a food source. Pick a single colony of E. coli OP50 bacteria from a fresh streak on LB agar and inoculate 3 ml of liquid LB broth. Culture in a 37°C shaker overnight. Prepare serial two-fold dilutions of the overnight culture. Then measure the absorbance of each dilution at 600 nanometers by spectrophotometer in triplicate. Further dilute each bacterial suspension 1 millionfold. Evenly seed 0.4 ml of bacterial suspension of each dilution on a 100 mm LB agar plate and incubate at 37°C for 18 to 24 hours. Count the number of bacterial colonies grown on each plate in order to calculate colony-forming units per milliliter for each sample. Now, plot OD600 as a function of bacterial concentration and perform a linear regression analysis to establish the relationship between measured OD600 values and OP50 bacteria concentrations.